So my laboratory focuses on two main topics. So the first one is embryonic development. So we want to understand how does an embryo develops from one single cell that results from fertilization. And we are particularly interested in the processes that create asymmetries within an embryo, meaning how do you create a left side of an embryo that is different from a right side. And why is this important? Because if we look at our body, we can really appreciate that we have asymmetric distribution of the internal organs. The heart on the left side, the heart on the left side and the liver on the right side. So this means that this asymmetric distribution of the organs has to be established during embryonic development, and it is very important for the function of an organism. On the other side, we are also interested in the processes that control symmetric development. So during embryonic development, there are territories that have to keep their symmetry. They can't be asymmetric, like the territories that are going to give rise to asymmetric structures like the skeleton and the skeletal muscle. So how is this coordinating during embryonic development is one of the focus of my lab. And to do this, we work on zebrafish, which is a fish that is a fantastic model to study these problems for a number of reasons. One of them is the fact that embryonic development occurs outside the mother's body, so we can really follow development in vivo, in front of our eyes. And then the second reason is because the embryos are very transparent, so we can see the organs being formed inside the body of this embryo. And then, of course, we have the molecular and the genetic tools to answer the questions. So we are also using the zebrafish to study regeneration. So it would be wonderful to be able to regenerate a heart or a liver after severe injury or chronic diseases. And so a process of regeneration can be seen as a recapitulation of development. So for a developmental biologist like myself, in a way, study regeneration is like a follow-up of what we know about embryonic development. And again, we use the zebrafish because the zebrafish is a fantastic model to study regeneration, because actually it is an organism that can regenerate all its organs. So it can regenerate the heart, it can regenerate the liver, it can regenerate the spinal cord. So we are now looking at the zebrafish and how it can regenerate, in this case, the spinal cord, in order to be able to understand how can we, in the future, be able to regenerate the spinal cord of a mammal and make, for instance, a mammal recover its motor function after a severe injury, which it doesn't by natural terms, let's say. So that's what we do. So I like a lot Lisbon. I'm not from Lisbon, but I've been living here for a long time and I really like it. I feel that Lisbon is changing quite a lot and very rapidly. So I like to really walk around and to discover the changes that Lisbon has to offer every day. And so I like to discover new little cafes or shops. And so I think that the thing that I like the most to do in Lisbon is really to walk around and discover what are the changes and what are the new things that are happening in Lisbon, because I feel that Lisbon is very vibrant in these days.